Myself Shraddha Shinde from FY MPharm Pharmacology of Siagat College of Pharmacy. Today I would like to discuss this paper, Drug Discovery in Cardiovascular Disease Identified by Text Mining and Data Analysis. So talking in brief about it, we could say that the paper describes the use of text mining and microarray data analysis to identify potential drugs for cardiovascular disease. The study identified a set of genes that could be targeted by existing drugs to expand drug indications for atherosclerosis related disease. In this, first, the method used over here is of text mining and microarray data analysis for obtaining a common set of genes. Next, it is followed by gene ontology and KEGG analysis, followed by the next pathway, that is protein-protein interaction network, and finally a significant gene cluster in the protein-protein interaction network is chosen to execute a gene-drug interaction analysis for finding out the potential drug. The results they have got in this study are 1788 text mined genes were found, where 275 differentially expressed genes were also obtained from the process of text mining and data analysis. 93 genes were duplicated between the text mined genes and the differentially expressed genes, in which 89 genes were upregulated once 4 genes were downregulated. 23 genes clustered in the significant gene module, and finally 8 genes out of 23 genes had the potential of targeting 20 existing drugs. To conclude, the finding of these 8 genes which were associated with inflammatory response, wherein they targeted 20 existing drugs and they had the potential, or they may target the existing drugs in atherosclerosis related disease. For the introduction, we can say that the paper highlights the prevalence of atherosclerotic cardiovascular disease as one of the most common cardiovascular diseases and a leading cause of death worldwide. The atherosclerosis cardiovascular disease has two primary forms, which is the ischemic heart disease and cerebrovascular disease. The occurrence of this has multiple intrinsic factors, including the accumulation of immune cells, production of pro-inflammatory cytokines, and dysfunction of endothelial and vascular smooth muscle cells. External risk factors of cardiovascular disease includes hypertension, hyperlipidemia, high cholesterol, diabetes, obesity, smoking, and unhealthy lifestyle. The method used over here for text mining and microarray data analysis for obtaining the potential drug targets for atherosclerosis related disease followed the method of firstly text mining. The author has used the open access website PubMed to Ensemble to perform the text mining. They have entered the keywords, in their case atherosclerosis, into the website, and they have accepted all the unduplicated genes which make up the text mining genes. Secondly, microarray data analysis was done, in which they have used microarray data set GSE 28829 to obtain differentially expressed genes related to atherosclerosis, followed by gene ontology and KEGG analysis. They have performed this process for genes and genome analysis on the text mined genes and differentially expressed genes to identify their biological functions and pathways, followed by protein-protein interaction network analysis, where the author has constructed a PPI network using the significant genes clustered in the PPI network to identify potential drug targets. The gene-drug interaction analysis author executed. The author has executed gene-drug interaction analysis on the significant genes clustered in the PPI network to identify potential drug targets for atherosclerosis related disease. Overall, the study used a combination of text mining and microarray data analysis to identify potential drug targets for atherosclerosis related diseases. So, this is the flow chart. Here, the text mining and the microarray data analysis combinedly found out genes that are they have got the 93 genes, of which common genes were the common genes between TMG and DEG. Further, it was processed for the gene ontology and KEGG analysis. From there, it has reached to protein-protein interaction analysis, where they have found 88 genes, and for the in the string domain, and then they have from the M code they have got significant gene module, that is 23 genes, and further going in the drug gene interaction, they have found about 20 drugs, and final 8 genes were obtained. This is all the statistics over here. Here, this image shows the PPI network construction and significant gene module analysis, wherein the first image is the entire PPI network of common genes, and the second figure shows the significant gene module including 23 genes. Here's the discussion and the comparison done with the other studies found out. So for the results, the paper has identified 8 genes. It's over here. That are associated with the inflammatory responses and which has the potential of targeting 20 existing drugs. These genes might be used for atherosclerosis related diseases as well as their original drug indications. Functional enrichment terms of these 8 genes were mainly involved in 11 gene ontology terms, with the most significant being the inflammatory response. So the study used text mining and microarray data analysis to identify these potential drug targets. To conclude the paper, there are text mining and microarray data analysis can be used to identify potential drug targets for atherosclerosis related diseases. The study identified 8 genes associated with the inflammatory response that can target 20 existing drugs. These findings may expand drug indications for atherosclerosis related diseases. About these charts, these are the drug and their genes, drug and gene interactions that are shown. All the genes and all the drugs associated for it are enlisted over here. The gene subtypes are also described.